Okay, our next speaker is Dr. Robbie MAJZNER. He is the director of the Pediatric and Young Adult Cancer Cell Therapy Program at Dana-Farber Cancer Institute and Boston Children's Hospital. He is focused on the development and deployment of immunotherapies for pediatric cancer. He received his MD from Harvard Medical School, followed by training in pediatrics at New York Presbyterian Columbia and pediatric hematology oncology at Johns Hopkins and the National Cancer Institute. His work in the laboratory centers on engineering platform technologies to improve the efficacy of CAR T cells in solid tumors and overcome therapeutic resistance. Concurrently, he is focused on translating laboratory advances in innovative clinical trials for children with incurable cancers. And his talk today is going to be targeting GD-2 with CARs and antibodies. Dr. MAJZNER.
Thanks, Pat, for the introduction. Nice to see everyone here today, and I'm going to talk a bit about a little bit of mix of history on targeting GD2 and what GD2 is, and some new science from the lab, but mixing that in with some information and thoughts about drug development for pediatric cancer, which I know is at the top of everyone's mind here. These are my disclosures, although none are directly relevant to what I'm going to speak about today. So GD2 is, there's a picture of GD2 on the right side of the screen there. It's what's called the glycolipid. It's basically a combination of a fat and a sugar, and it sticks outside the cell. So things can recognize it, like antibodies, immunotherapies, basically see this sugar sitting outside the cell. And it was actually one of the first cancer antigens discovered. So if we think about immunotherapies, we need a way to get the immune cells to say, that cell is not normal, and we should attack it as opposed to the normal cells. And two groups, one on the West Coast and one on the East Coast, kind of simultaneously discovered that this is on many different types of cancer. And it's interesting that it was really one of the first targets ever discovered, and it's just got an amazing history because they were hoping to develop it for many different types of adult malignancies where it really didn't have a development path, and I'll talk about that, but that it's just become this, like, the only really validated target we have, not only in neuroblastoma, but in any pediatric solid tumor. So the first, at the time that GD2 was discovered was also the time that people were figuring out how to make antibodies. And you can think of an antibody basically as a flag that basically will bind to its target, here to GD2, and then flag that cell for removal by the immune system. So it basically kind of passively tells the immune cells that are already in the patient's body, come here and kill that cell. And you want it to bind to the cancer cell and not to the normal cell. So I mentioned two groups. One in California really started making what eventually became dinutuximab, or was CH1418 at the time, and then Nikon Chung's group, which eventually ended up at MSK, made 3F8, and these are what have become dinutuximab and naxitamab in modern times. But basically, they figured out, okay, this drug, which people were interested in because GD2 was expressed on melanoma, also works for neuroblastoma. And this is just some mouse models, and the specifics don't really matter there. But they saw preclinical data, and they were able to get neuroblastoma patients treated in the first clinical trials of these agents. And so this is results from a trial that started in the early 1990s where they gave only the antibody, and you can see there's a bunch of patients, sorry, I don't know if the pointer will work, bunch of patients that have melanoma, a few that have osteosarcoma, and then a scattered patients that have neuroblastoma. And the only responses they saw in the whole trial were in neuroblastoma. So that's great for children and for neuroblastoma. It's not so good for drug development because it is, as you guys know, a very small population and very difficult to develop drugs. And the reason that you see differences in response may have to do with the amount of GD2 on the cancer cell. So the more of the sugar that's there, the more the antibody can come and bind to the cancer cell and flag it for removal by the immune system. It actually turns out that neuroblastoma has a ton of this sugar, this GD-2, whereas other cancers, that's a lung cancer I show, have very little. The brown is how much of the cancer there is. Then to make matters even worse, this thing wasn't gangbusters on its own in neuroblastoma. So if you had a patient that had relapsed neuroblastoma and they had, soft tissue disease in the bones, bone marrow, but also in lymph nodes or masses in the belly or anywhere else, the antibody didn't really work to regress those big masses of disease. It only worked in places where there were small amounts of disease, let's say in the bone and bone marrow. So it was an even harder development path because it wasn't like you could give it to patients that had relapsed neuroblastoma and then they would go into remission. So proving the benefit became really, really difficult. And this was true both of CH1418, which became dinutuximab, but also the antibody 3F8 that was being developed at Sloan Kettering. So both antibodies only worked when there was only a small amount of disease. So the Children's Oncology Group, funded by the NCI, and I think Sarah spoke about this trial a little bit with you, actually ran a randomized clinical trial for patients that were at the end of therapy. So they had finished all of that chemotherapy, radiation, and surgery, and then randomized patients to either get, isotretinoin, which is retinoic acid, alone, or retinoic acid plus the antibody. And this was a really, really important study because it showed this 20% increase or 20% decrease in relapse in patients. And this is what led to the FDA approval of dinutuximab. And eventually, after that entire randomized trial was run and paid for by the NIH, that drug basically got licensed to United Therapeutics. And it was only once it was proven and shown that it would be FDA approved that United Therapeutics said, okay, now we'll take this on. So they no longer had to bear the risk of developing it. All they had to do was really manufacture it, and now they are able to sell it. And, keeping up a stable drug supply is not a trivial thing, and it's what drug companies are really good at. But I want you guys just to understand for the drug company, unless there's a huge market at the end, oftentimes the risk benefit of having to invest in these types of trials, which are extremely expensive, it's not there for them. And that's why generally, it's not that they don't want to help kids with cancer, but it generally, the market, for a private company isn't working for our patients to do this kind of drug development. But fortunately, really on the backs of pediatric oncologists, scientists, academicians, the NIH, and then a lot of philanthropy driven by families, these drugs were able to get approved. And what this has allowed, I think Sarah talked about a little bit, is actually to use these drugs in new ways. So this was the trial where they randomized immunotherapy versus a different type of chemotherapy and really found for the first time that there's an amazing response rate for patients that have relapsed disease. And then the work at St. Jude also using this in the upfront setting that now has led to the current COG trial studying upfront chemoimmunotherapy. And there is a direct line from the decision of pediatric oncologists to get this drug FDA approved that allowed for the discovery of chemoimmunotherapy for refractory neuroblastoma. It's only because the drug was approved that then they were able to do a study in this type of setting. And otherwise, it really wouldn't have happened. And now, hopefully, we'll see positive results using this in the upfront setting in this trial that will lead to using it even more widely. And the same thing can be said for naxitamab, Danyelza. It was really only after this drug, this is a humanized version of 3F8, was proven to have efficacy within Sloan Kettering, that then they were able to attract investment from biopharma. And there was a startup, or what was a startup, now is an established company with an approved drug, Y-mAbs, which then brought this over the finish line by running a registrational study. And the registration was done a little differently. Things have changed with FDA since the early 2000s. In certain cases, when you can show regression of disease, from patients, so you can take patients that have real disease and then give them a medicine and show that it goes away and stays away on scans, you can get a certain type of FDA approval, and that's ultimately what they were able to do with DANYELZA. But as you guys may know, this is actually only approved for patients that have bone or bone marrow disease, and not for patients that have soft tissue disease. The specifics of that don't really matter. You actually can use it off-label in any way that you want. But the important thing that I want to mention here is the investment from the drug company or from the startup only came after the drug was clinically de-risked within an academic environment at Sloan Kettering. And I think that's just another example, just like dinutuximab, of how much work pediatric oncologists and their partners in philanthropy and families need to do in order to carry these drugs to approval. So I'm going to talk a little bit about the science that we do in my lab and then try to weave in some more of this talk about pediatric drug development. So we have this anti-GD2 antibody, but unfortunately cancer is smarter than most of our therapies and finds ways around all of our different therapies. It finds ways around chemotherapy and it can find ways around immunotherapy. So this is data from patients that were treated with anti-GD2 antibody, and I show 4 patients there on the left side, and this is the amount of GD2 that's on their surface. So sorry, I don't know if the pointer is working there, but you see this one is high, this one is high, this one it's very heterogeneous, it got high and low, and this one it's nearly absent. So all 4 of these patients had previously seen anti-GD2 antibody, and some of those patients had lost or downregulated it. This picture is not such a good picture, but this is another patient who had seen anti-GD2 antibody. Where it's brown, it means there's GD2, and you can see some places are positive and some places are negative. So the cancer is figuring out how to basically reprogram or reorganize itself to reduce its amount of GD2 so the antibody doesn't work as well. So this is how GD2 is made, and even for me, who I run a research lab and went to medical school, this is a nightmare for me to look at because there's so many steps and it's such a complicated process. Generally, here's GD2 here, and people have always looked at this gene or this protein as the last step to make GD2. So they've looked at tumors and said, okay, if it has that, it probably has GD2. I'm going to show a little bit of science here, but I'm going to do it and try to talk about it in a easy to digest way. It actually turns out that enzyme that we thought was going to be involved when GD2 was lost was not the enzyme. It was one step before that, this other one. Instead of this B4 one, it's this ST8 one. So that was surprising. We never would have known if we didn't start looking at both patient samples and samples from mice. But we found that GD2 was going away, and it was going away through loss of one of those steps in that really complicated process. Now, with the help of the people I showed on the last slide, the lab figured out that actually you can reverse this. It happens through something that happens in the cell where the cell figures out how to turn off this one specific step in making GD2, and there happens to be, an FDA-approved drug that can reverse that. It's called an epigenetic inhibitor. This is approved for a type of adult lymphoma and then a type of a rare sarcoma. And you can see here, if you have GD2 low and you treat the mice with the drug, the amount of GD2 goes up 800-fold. So this is awesome because there's an FDA-approved drug, and we said, okay, let's mix this with dinutuximab, and you see here, dinutuximab by itself has no efficacy, so you see tumor in the mice here. Tazemetostat really does nothing, but if you mix them, you start seeing an ability for the antibody to work again, even when the tumor cells have lost GD2. So we were excited about this and said, okay, let's now talk to the company that has an FDA-approved drug. The drug's actually already been in pediatrics because there's other diseases that people have wanted to use it for. And let's see if we can run a clinical trial combining it with the anti-GD2 antibody or chemoimmunotherapy. So we applied to them 3 separate times. The company was a small biotech, eventually got sold to a bigger pharma company. And then this is the response they gave to our last LOI that we sent them, which is a letter you write to say we're interested in using their drug. We weren't asking them for money, we were just asking them for the drug. But they wrote, I'm writing regarding your submission on tazemetostat. After careful evaluation, the committee has unfortunately decided we cannot support this submission at the time as our focus is on the adult patient population. So even here, they've got an approved drug. It doesn't actually cost that much to make. They've got a constant supply, and they're just like, yeah, we're just not going to do the trial with you. So this, unfortunately, has not gone forward, although there are people interested in still doing this. We just haven't really figured out a way to do it. So we said, okay, we got to work around them. We got to find another way to deal with this problem of GD2 loss. So this is that kind of that pathway I showed you before, simplified. So GD2, as you can see, is down here on the bottom, and we figured out that it's this enzyme that you lose when you lose GD2. And what that's going to do is basically going to cut out this side of the pathway. So you can no longer convert GM3 to GD3. The specifics don't matter, but basically you're stuck over here. So you still have lots of, these are all sugars, you have lots of this sugar, but this enzyme, this protein, still is there. And so we said we probably are going to get, when we lose GD2, we're going to get an increase in GM2, kind of like a seesaw. And that's exactly what happened. This is some mouse data where you lose GD2. You see it's high in black on the mice, but when you treat them, they lose GD2. This is with different GD2 immunotherapies, but GM2 goes up. You can see that on the right side. So it's kind of like you try to, now if you lose one target, you might have another, and you can play a game of kind of whack-a-mole with the cancer. So that's exactly what happens in patient samples. Also, all of the samples, even when they lose GD2, we find high GM2. So we said, okay, this may be an alternative target. And GM2 was also discovered in the 1980s, around the same time as GD2. There were studies of GM2 antibodies in adult cancer, in lung cancer and myeloma. They ended up not going forward, probably because the levels weren't that high. I'm going to skip that. But we said, okay, maybe we could instead target GD2 and GM2, or only GM2. And so these are 3 mouse models. The specifics don't really matter, but you can see in this mouse model, GD2 doesn't work, and GM2, actually, you get some efficacy. So this is the amount of tumor, and it takes longer for the tumor to grow out if they got GM2. This is another one, same thing. And then this is a third one where I think the result is the most impressive. You can see either GD2 or GM2 antibody controls the tumor for a little bit, but when you combine the antibodies, you actually are able to get rid of the tumor completely. So either antibody drives resistance one way or the other. The GD2 pushes GM2 up, the GM2 pushes GD2 up, but when you treat with 2 antibodies, you basically can trap the cancer. Okay, so this is more stuff about drug development. I'll just say this is a problem. I mean, Sarah mentioned it also when she was speaking. We run into all the time. Drug companies generally, unless they're really compelled to, most of the time they're not that interested in dealing with pediatric trials. And so we previously ran a trial of a combination of GD2 with another antibody. And when we did that trial, we actually had a lot of trouble getting the anti-GD2 antibody from United Therapeutics. So nixitamab hadn't been approved yet. United Therapeutics was the only source in the US of anti-GD2 antibody. And they told us, even though it was pediatric oncologists and the NCI who had paid for the trial that led to the registration of their drug, they told us, oh, we're studying this drug in lung cancer right now. If it ends up positive, we're not going to have enough drug for you. Ultimately, the NCI was involved in this trial and called them and told them, you're crazy, you're giving us the drug, and they sorted it out. But I think this is just a recurring problem. Sorry, there's the old slide, so this is the same thing I showed you before. But so, you know, my lab really focuses on CAR T cells because we view it as something that we can do completely within academia and not worry about getting a drug from a drug company. So I'm going to talk for a minute about how CAR T cells are made. This is an example from leukemia, where basically a patient that has leukemia, you can take their T cells. These are cells that can kill cancer cells, take them out of the body. You can reprogram them with a virus to express what's called a CAR. Sorry, I meant to point. What's called a CAR, and it's basically, you just like express an antibody on the surface of the T cell. So you basically combine the T cell's ability to kill with the antibody. And when you put the cells back in the patient, you basically go, they stick to the cancer cells, and then they can kill them. And this has worked incredibly well in leukemia. This is Emily Whitehead, the first child who ever got CD19 CAR T cells. She's way more than 10 years cancer-free now, and actually she was treated at CHOP, and now she's an undergraduate at University of Pennsylvania. It's an amazing story. Also, another drug that was initially completely developed within academia, and then only once it actually had proven efficacy, Novartis picked up this drug. A few others were being developed at the same time, and ultimately developed it both in adult lymphoma and in pediatric ALL. Skip that. So this is a schematic of what a CAR is. You have the antibody, that flag that I showed you before that you put in patients, and it tags cells for removal cancer cells for removal by the immune system. And then you have a T cell, which has a much more complicated way of killing through this thing called the T cell receptor, but is the most potent killing machine. But T cells don't recognize antibodies. And then this Israeli guy in 1989 had this crazy idea to combine an antibody through genetic engineering with the signaling domains from a T cell, and made what's basically this Frankenstein receptor that puts an antibody on the outside of a T cell and is able to harness that killing power to recognize whatever target you want. And so you could do that, for instance, for GD-2. So this is our new model of drug development. Here's that company that didn't want to give us tazemetostat. Instead, we take our child, we get them into the car, we treat them. They drive right by and wave, and then when they grow up, they really tell the company what they think. Yeah. Of course, I'm being tongue-in-cheek about this. I'm also involved in biotech companies, and really, they are a very important part of the ecosystem. But really, the way the market works, it really is not driven in a way or designed in a way to drive therapy development in pediatric oncology. So back to this story about losing GD2. We did the same thing. In addition to using an antibody combination, we also did a CAR T cell combination. So this is a tumor that we put in mice, and you can see at baseline, you can just look at these pictures. It expresses a lot of GD2 and has no GM2. If we treat with the GD2 CAR, the tumor grows out, so we don't cure the mice. The GD2 is gone, and now they express GM2. If we treat with the GM2 CAR, it doesn't do anything because there's not that much GM2 there to start, but you see that the GD2 goes up. Sorry, I can't point, so I'll point on that one. GD2 is really high, and GM2 is gone. And then we made this CAR that can recognize both at the same time. So this is cool. You can make a new drug in your lab. You just see some problem, and you can just design this. I mean, it probably sounds very complicated to you guys, but literally you're like, oh, I know the sequence. You enter it into a computer, and you order it from this company that either works in New Jersey or China. I'm not sure anymore where they are. And then a week later, you get this DNA cassette, and then you can just put it into your T cells. It's crazy, like science fiction. But we designed that, and then you can see it cures the mice completely of their tumor. So really cool that understanding the biology of how GD2 is regulated allows us to make new therapies. And I just want to tell you that GD2 CARs are coming, and they're actually kind of already here. And this is a story that started in the early 2000s at Baylor, Texas Children's Hospital. Actually, the first CAR T cell that was ever in a child was a GD2 CAR, long before CD19 CAR for leukemia at all. This was a trial where they took the antibody portion of dinutuximab and connected it to the signaling domains of a CAR T cell. And even back then, in the mid-2000s, when none of us had heard of CAR T cells, they had some responses. There were limited responses, but 3 patients had a complete response. There were patients that had very small amounts of disease. One of those patients is still living today, 18 years later, which is really pretty incredible. But the results weren't good enough, so they tried to iterate. They added some more engineering things, so they redesigned the CAR. You can see now there's this red thing and this green thing here. These are extra signaling domains. They said, okay, let's try to do it. Unfortunately, they actually had no responses, so they made it worse when they added these things. That trial was run in the 2010 era, then they had to go back to the drawing board. But then there was this group in Italy, and some of the families here may have heard of this group in Italy who actually had all trained in Texas, took it back home and changed the construct a little bit. And they didn't change it in any way that's rocket science. Like, they just swapped out one of those little signaling domains. Again, something you can do relatively easily, like from the safety of your own bed. Actually, it worked incredibly well. So they ended up having a 60% response rate, half of which were complete responses, half of which were partial responses. There's caveats to the data. Only 50% of the patients on the trial had ever seen an anti-GD2 antibody, which may push down GD2. None of them had actually seen chemoimmunotherapy because it wasn't available in many European countries at that time. But there is no doubt that the thing is active, and especially in patients that have very low tumor burden. And they're trying now to figure out how to explore it, to use it in patients that are kind of at the end of therapy and maybe have just a small amount of disease, and see if you can consolidate and keep that patient in remission. This has been really important actually for CAR T cells in general, because it's the largest series of responses to any CAR T cell in any solid tumor anywhere in the world for any cancer of people of any age. So this is really pediatric oncologists, again, leading the way and teaching the world how to treat solid tumors. And we're hopeful that this continues to develop. But point I want to make here is I showed you guys example of 3 different trials for GD2 CAR evolution. There have actually been a few more in between also. It's basically 20 years of development in order to find one clinically active drug. And this is done really in a very serial fashion. So you do one trial, you finish it, try something else, and in between you got to fundraise to get, a trial costs about $4 million to run between all the costs for making the cells and carrying out the trial. So a lot of fundraising and grant writing that happen in between. But, the technology for this CAR3 is not that different than the technology for this CAR1. Like, by the time this trial was done, all of the technology existed for CAR3. So we need to be doing more of these trials at more centers, more quickly, and coordinating our ideas to figure out what is going to work to make these therapies more effective. And we can do it in a way that we really can't do it when we're going out with drug companies to develop drugs, because we're dependent on how they design the drugs for whatever whatever indication they're going after, whether that be in lung cancer or melanoma. So just to conclude, I hope I gave a little history about GD2. I think it's just amazing that it was one of the first cancer targets ever discovered, and it really would have died with many other cancer targets that have failed in adult oncology if it weren't for pediatric oncologists, and it wasn't me. So people that ran all of those trials, both through Children's Oncology Group and at MSK, and just really kept pushing it. I think we need to do a better job as immunotherapists. So my lab mostly focuses on the design of immunotherapies, but instead in understanding how the biology of the cancer can drive our response to immunotherapy. So we found this simple thing of we were able to find the protein or the enzyme that goes down in order to make GD2 disappear, and this allows us to find GM2 as this kind of whack-a-mole type situation. Drug development for kids is really, really tough for these very small indications, especially as, thank God, survival rates are really improving for neuroblastoma nowadays. So if survival rate is as that gets better, the addressable population shrinks and companies become even less interested in that. It's really been by clinically de-risking things in academia, previously with antibodies, and now I hope with CAR T cells that we've actually been able to see advances. So I'll stop there and take any questions.
Thank you. Anyone have any questions? No? Okay, go ahead.
So I am actually curious about this FDA-approved drug. I'm not going to try and say the name.
Tazemetostat.
Thank you. they didn't want to use it they wanted to focus on adult patients. So have you ever thought about using it in adult neuroblastoma patients? And the reason I ask is because I have one.
It's a great question. I think I think when they gave that answer, mostly what they were saying is they don't want to run the trial because even if it works for adult neuroblastoma patients, right, that's even more rare. The drug is actually FDA approved, so we can give it to children. And actually, I have a few colleagues that have tried this approach. You know, it's very hard to get an idea if something's working if it's not in the context of a clinical trial.
Okay, that was going to be my follow-up question. Can it be given as, like, when it's not part of a trial? So because my child is an adult, technically,
Yeah.
I could potentially find somebody who might want to try it if that were appropriate.
Yes, and I mean, I gave it to one patient when I was at Stanford. I have at least one colleague who's given it. You know, the problem is it's almost impossible to know if it matters until you run the right kind of clinical trial. It also, you don't only really should be giving it to a patient where you think GD2 loss is the reason that this isn't working, and that's almost impossible to prove nowadays.
Right.
Because it is very hard to assess GD2 in kind of clinical laboratories because you can't do it through the typical methods, and we've had to do a lot to set up being able to assess it.
Okay, yeah, that's fair.
Okay, another question?
I just want to say thank you for showing this. I'm also obviously very thankful for anti-GD2 therapies. I mean, we've had a lot of it for ourselves. This is something that it's just really eye-opening to me to know that the GM2 goes up, and there's also a drug that can bring the GD2 back. Because it feels like there's a lot of the newer therapies, like you mentioned, the CAR T stuff, that is targeting GD2. So I guess I know you said that it's pretty easy to do this in a lab. Do you think there's any long-term runway for a GM2 antibody? Or do you think that's kind of a pipe dream kind of thing?
So, you know, I'd like to think so. So first of all, these are laboratory experiments, and they, as you guys know, they don't always translate. Like we've had things that we've done in the lab, and they look great, and then you go into patients, and they don't work, and we don't always know why. For the antibody, that specific antibody is in a Japanese pharma company that has had it on the shelf for 15 years. And could they be convinced to take it off the shelf and try it in a trial? It's possible. From my perspective, you know, within what I do within my lab and clinical program, we are planning to eventually, hopefully, have a combined GD2 and GM2 CAR T cell program where we try to target both of them at the same time. That's not to say CARs are the best way to do this. You know, if upfront chemoimmunotherapy gets cure rate up to 80%, and then like actually it turns out that you can chip another 10% off or increase another 10% by adding a GM2 antibody to the end of therapy, I think that would be great. It's just a huge challenge. If we had proof of concept that this is happening and that you might be able to overcome this with CAR T, you might be able to get more interest on the antibody. I don't think it's impossible, but I also think for me, I think probably we're going to focus on doing the CAR to try to get some clinical proof of concept earlier.
Well, it's exciting work. Thank you for doing it.
Go ahead, another question?
Can you tell us some of the downsides of CAR T therapy and, I don't know, some of the risks involved?
Sure. So first, normal tissue expression of the antigen. So if the target is on the cancer cell, the T cells will go kill it. But if the target is on a normal cell, the T cells could potentially go and kill it. And we see that with CD19 CAR in leukemia. So CD19 is expressed on all your normal B cells. And the patients that get this, they go into remission from their leukemia, but they lose their normal B cells. Fortunately, you can live without B cells, so that's expendable. With GD2, it's expressed, as you guys know, on nerve cells, and patients have very bad pain when they get GD2 antibody. Actually, We don't understand why, but patients do not have pain and have not had thus far killing of their nerve cells, also the brain expresses GD2, from GD2 CAR. And it's somewhat a mystery why. We think it may be because the amount of GD2 is so much higher on the cancer than on the normal nerve cells. Actually, thank God that group in Baylor did that clinical trial in the mid-2000s. I think it was probably a different time from a regulatory perspective then. They actually didn't know that it wouldn't cause pain or cause neurotoxicity, but they were able to bring it forward, and that's led to all of these further developments. So that's one risk. And one day we may have a GD2 CAR that's good enough that it has amazing results with curing the cancer, but also causes major permanent damage to nerves. Right now, we haven't seen that. They seem to be in a therapeutic window where they don't touch the nerves, maybe because they have lower levels of GD2, but they kill the cancer cells. The next thing is when patients get CAR T cells, it's basically you get like a huge immune response in the body, kind of like when a patient has sepsis or a really bad infection. You have so much going on, the T cells start seeing the tumor and they start killing it, that it releases chemicals and proteins that drop the blood pressure. Patients end up in the ICU. Patients end up with high fever. Around CD19 CAR, this was a huge challenge when they started, but they've learned to manage it really well, and most patients actually do quite well. In many places, this is done as an outpatient medicine now. And you know, I think those are the major risks from a clinical perspective. From a drug availability perspective, you need to go to a center that does it. You have to have your T cells taken out of the body. They have to go through a biologic process that introduces this chimeric receptor. And sometimes that process fails. Sometimes we make CAR T cells for a patient, or we try to, and we're unsuccessful. That patient has gone through a lot to get on that trial, to oftentimes traveling, giving their cells, et cetera. So it's not an off-the-shelf product like, for instance, an antibody is. So I think, you know, those are probably the major downsides to CAR T cell therapy.
Thanks for explaining that. I had one more question. Could CAR T therapy ever theoretically, I know you'd have to try it, be done with chemotherapy?
Yeah. So patients do get a small amount of chemotherapy before they get the CAR T cells, and that's mostly about eliminating blood cells that might otherwise reject the CAR T cells when they're given. Generally, T cells, you know, don't like chemotherapy. It's why we have infections after periods of chemotherapy. But people have done things like, because you take the T cells out and you can reprogram however you want, you can make them resistant to all types of drugs also. You give them like a resistance gene that someone finds in cancer, and they can program that into the T cell, so it's resistant to a chemotherapy or a targeted agent, for instance. And so people are doing things like that. I think in the short term, like what they're trying to do in Rome right now, or planning to do in Rome, is try to see if patients, after they get standard therapy, so that means still going through the whole thing, all that upfront toxic therapy that patients get right now, giving this at the end, kind of like the antibody is given at the end, trying to kind of sop up anything that's left there. And I think in the short term, we'll either see it being used for patients with frank relapse or that type of situation. You know, it's our dream that eventually we're able to pull back from the amount of chemotherapy that's used and integrate this into an upfront type of therapy. But I don't think we're there yet.
Thank you so much.
Anything else? Anyone else? Okay. Thank you, Robbie.